Okay. Well, welcome everyone to the Spring Science of Information seminar series. My name is Brent Ladd. I'm the Director of Education for the Center for Science of Information that we're based at Purdue University, but we collaborate with 10 or 11 other university partners around the country. And we're part of the National Science Foundation's STC program. And today I'm delighted to introduce Lina Aboulmouna and Rubesh Raja, who are both center members in the Department of Chemical Engineering at Purdue, and they work in the Ramkrishna Lab there. And they've both been engaged in, I think, a unique long-term collaborative research effort with the Subramaniam Bioengineering Lab at UC San Diego, which is also one of our institutional partners. So Lina, I know I've just been talking with her that she's anticipating graduating this August with her doctorate. And Rubesh is a postdoctoral research associate that also joined the center in the last year or so, also working on this unique collaboration. So we're really pleased to have these two young scholars present today in the seminar. And just one housekeeping note, if you haven't used Zoom before and you have a question, just press down and hold your space bar on your laptop and you can speak a question, or you can type into the chat function, and I think it's at the bottom of the screen for most people. And I'll try to monitor that if you have questions as we go. So thanks for joining today, and I'm going to turn it over to Lina and Rubesh.
Thank you, Brent, for coordinating this lecture series. So good afternoon, everyone, at least for those on the East Coast. My name is Lina, and I'm going to be co-presenting with my colleague, Rubesh. And as mentioned, this work is done in collaboration. So this work is done in the Ramkrishna group at Purdue in collaboration with the Subramaniam group at UC San Diego. So we'll be highlighting the work on a cybernetic modeling approach of eicosanoid metabolism in bone marrow-derived macrophage cells. So let's start out by taking a look at our body's response to an injury. The body has a two-line defense system against pathogens or germs that can make us sick. Pathogens include anything from bacteria, viruses, toxins, parasites, and fungi. The skin shown in the image right here is the largest organ of our bodies and acts as a barrier between invaders, which are these pathogens, and the body. The body's second line of defense, the immune system, triggers an inflammatory response when there are foreign invaders to fight off. Some of these symptoms of inflammation can include pain, redness, swelling that is sometimes warm to the touch, stiffness, and loss of function at the site of inflammation. Our immune systems kick in during viral infections as well. We've been hearing quite a bit about COVID-19 lately in the news. So here's an overview of what the virus does to our body. The virus can enter the body generally through the mouth or the nose, and from there, the virus shown here can make its way down into the air sacs inside of our lungs, known as alveoli. Once in the alveoli, the virus uses its distinctive spike proteins to hijack the cells. The primary genetic programming of any virus is to make copies of itself, and COVID-19 does just that. So once the virus RNA has entered a cell, new copies are made and the cell is killed in the process, which releases new virus to infect neighboring cells in the alveoli. During a severe viral infection, there are multiple cells that are activated in response to the virus, which then release chemokines to attract more inflammatory cells to migrate from the blood vessel into the site of inflammation. And these cells release additional chemokines and cytokines to amplify what you've likely heard of as cytokine storm. As you can see, the inflammatory process is rather complex and involves multiple key players, but we're going to focus on one component of the inflammatory process, which are these macrophage cells. The macrophage cell is known as the body's first responder to a site of inflammation. In a controlled laboratory setting, there are extensive studies that have been done in order to mimic the physiological response of bacterial infection on macrophage cells through exposure of factors like lipopolysaccharide, or LPS, shown here, which is found on the surface of bacterial cells. LPS is the bacterial molecule that specifically induces an inflammatory response in macrophage cells, and increased levels of adenosine triphosphate, or ATP, which is an energy molecule, act as a danger signal in response to cell death in the surrounding environment. Both of these initiate a cascade of events, which leads to the activation of the COX enzyme. And we're considering lipid metabolism that starts at arachidonic acid because the lipid molecules of inflammation are these prostaglandins. They're involved in transcription factors and regulation that give rise to molecules that act as markers of inflammation.
Typically, there are two different types of biological modeling approach. The first one, the first type is the black box approach, or some machine learning type modeling, where the box is hidden. So the availability of vast amount of biological data, including the experimental data as well as the omic data, has allowed researchers to train these models and predict different treatment as well as disease outcomes. But understanding the inherent mechanism is not possible with this approach. Another approach is an open box approach, where we try to mechanistically model different parts of the reaction network. It's not always simple to understand the vast amount of biology involved and the regulation happening in these cells. So now a third approach is we call a cybernetic model. Professor Ramkrishna and his group has developed this model to in order to tackle the limitations present in the other two models. The model framework describes gene regulation by attributing metabolic preferences to a suitable survival goal of an organism. For example, consider an enzymatic reactions. Most reactions in biological systems are enzymatic, which gives easy flexibility for the organism to control it. Here, the product PI is produced from the substrate Si in the presence of enzyme EI. The rate of production of P is written as follows, where the Ki EI is the production, Si is the production, and Gi Pi is the degradation. And the enzyme is also produced in the presence of the substrate at the rate KEI and degraded at the rate constant beta EI. So, cybernetics believe that the rate constants are not actually constant, but can be regulated by the organisms. So in order to incorporate the regulation, we introduce two cybernetic control variables, Vi and Ui, in these rate reactions. Here, the Ki and the Kei are the unregulated reaction rate constants, whereas the Vi and Ui induce regulation into these rates and can vary between 0 and 1. Here, the Vi is represented as an activity control. It can depend on the level of activity on an enzyme. And the Ui represents the synthesis control, and the bottlenecks in the transcription and translation mechanism where the enzyme gets created affects the Ui parameter. Though biologically these parameters have separate definitions, cybernetics tries to explain these, formulate these parameters using a simple goal-oriented approach. So the goal of any organism is its survival. In other words, evolutionary selection happens to organisms only when it's optimizing for its survival. In order to understand how the survival can be important, an important trait is speed. The more fast an organism is able to improve itself, you can you can see like the organism is able to grow faster. So, for example, a specific goal in the case of bacterial systems is to maximize biomass production rate within available resources. Here, by solving the optimization problem, we can get the expressions for the VI and UI in terms of rows, which is the return of investment. In this simple formulation, the return of investment can be just the unregulated rate constant with for the formation of product PI from the unregulated rate from the product PI to SA. So different biological phenomena can be explained by cybernetic models. So for example, dioxic growth. When E. coli is presented with two different substitutable food sources, such as glucose and lactose, it preferentially utilizes glucose first, followed by lactose. The following curve here, plot here shows the green curve represents the concentration of glucose, and the blue curve represents concentration of lactose, and the red represents the density of the bacteria in the system. So as you can see, the glucose is first utilized and allowing the bacteria to grow, followed by utilization of lactose and leading to further growth. Why glucose is utilized first is because the growth, if you take the slope here, the growth at which the bacteria grows is faster in the case of glucose than in the case of lactose. That's why a single optimization function which maximizes the growth rate will be able to capture this curve. And this cybernetic model has been defined by Kompala et al. Cybernetic models for simple bacterial systems are possible, but in order to understand complex system, we should think about the overall goal of an organism. So here, the overall goal of an organism is survival, but not for the individual cells. So the individual cells goal is not to survive, but to do its functions. Using that as a information, we are trying to find the cybernetic goal for macrophage cells. Here, the macrophage cell is a versatile, complex cell, and the goals can vary with time. But in the presence of these kind of external stimuli, we believe the goal of the macrophage cell is to maximize inflammation. Using this formulation as a cybernetic goal, Lina will explain the network involved in the Eicosanoid pathway.
So here's a more extensive network schematic. So although this is still only a fraction of the larger inflammatory response, we're still seeing like an incorporation of cell regulatory mechanisms for Eicosanoid metabolism and signaling using the lipid pathways that are derived from a database and literature. So in this model, we're taking a look at branches in the network in relation to key signaling mechanisms involved in inflammation. As Rubesh just mentioned, the defining feature of cybernetic modeling is in its dynamic control. So we need to define some end goal for our system. In the case of the macrophage response to inflammation, we have two objectives. We have cytokines, which are a small secreted protein released by cells and has a specific effect on the interactions and communications between cells, which is linked to this prostaglandin branch in green of the network. And then we have chemokines, which play a vital role in the induction of cell movement in response to some chemical gradient associated with the leukotriene branch, or shown in orange here in the network. Before we dive into some of the details, let's break the network into manageable chunks. So here's a bare bones of the network, starting with arachidonic acid that leads to downstream products. So for the reactions directly surrounding arachidonic acid, we apply standard kinetic equations to describe this framework. In the branching that leads to downstream prostaglandin and thromboxane products, we again apply standard kinetics, but here we include the cybernetic regulatory variables, U and V. Again, for the branching that leads to the leukotrienes, we apply the cybernetic framework with these U and V variables as well, but this is going to be associated with a different objective. But before we, go into that, where exactly does the cybernetic goal come into play in these equations, and how do we calculate the U and V? So I'm going to focus on this red, which highlights the branches of the network for illustration purposes of defining the cybernetic goal and calculating the return on investment rho. So we focus on the prostaglandins here because prostaglandins are well characterized for their roles in the inflammatory response. We look at regulation of prostaglandin synthesis in relation to a specific cytokine, TNF-alpha, which is a representative marker of inflammation and is our model's overall goal for this part of the branch. To quantify the relationship between the prostaglandins and TNF-alpha, we used a simple linear relationship of TNF-alpha levels as a function of PGI levels using their time series data. To determine the contribution of each PGI pathway leading to TNF-alpha production, we define associated weights, WI, of each pathway. The cybernetic variables compare the return on investment rho for each pathway and invest in enzyme production accordingly. This return on investment is used to calculate the cybernetic control variables, which regulate the enzymatic activity and synthesis in the cell. Now I'm going to describe the experimental setup which we use to try to model in this system. We have a control case in which no COX activation is happening. We have a treatment case which induces a calcium ion flux into the cell and activates the COX enzyme. And then we have a combined treatment of ATP and KLA in this cell, which activates the COX. So KLA here, which is katiolipid A, is a chemically defined substructure of lipopolysaccharide, or LPS, which I mentioned earlier is the bacterial molecule that specifically induces the inflammatory response in macrophage cells. Samples were collected at the different time points listed here on this timeline. So regulation of arachidonic acid is pretty complex, and since we don't have data on the upper part of this network, we simply used arachidonic acid as the initial input to the model and only simulated downstream products. So you can see here the mean experimental data, which is shown in circles with their associated standard error of the mean, are plotted from the 3 replicate experiments for the ATP stimulated case, which is shown in green, and the control case, which is shown in red. Our model simulation results are shown in this solid green and red lines. So visually, you can see that our model effectively describes the conversion of arachidonic acid into the downstream prostaglandin products. We went on to test the validity of our model using a third independent data set, which was using the combined ATP and KLA case. We used the parameter values that were obtained from our previous model simulation that I showed to predict the results of the combined treatment case. So again, the experimental data, which are shown in circles for the treatment case, are in the color magenta, and the control case, again, is shown in red. The production yields a good fit. The prediction case yields a pretty good fit to the experimental data, which further validates our model. So in addition to this qualitative measure that we use to see how effective our modeling is, we performed statistical tests to determine quantitatively the effective fit of our model and found our fit to, in fact, be statistically significant. And we also checked the biological relevance of our parameters by comparing the effective rate constants associated with the enzymes and their corresponding values that are reported in the literature. But for the sake of time, I've left a lot of that out. So we can extend this approach to the expanded network model that we talked about and include more detailed information. The value in understanding the network and its regulation is key in being able to resolve the unwanted side effects like pain and swelling associated with inflammation, while still allowing for resolution of infection. An example of this being critical is in the news that surrounding anecdotal evidence from physicians in Europe regarding the impact that NSAIDs have on the progression of COVID-19 in otherwise healthy patients. As of yet, there's not a clear understanding of the dysregulation that takes place in inflammation, nor any significant scientific studies to showcase this. So NSAIDs are nonsteroidal anti-inflammatory drugs that suppress the COX enzyme. Some of these NSAIDs include aspirin and ibuprofen, which are common over-the-counter drugs we're all familiar with, and they are non-selective COX inhibitors. An anti-inflammatory intervention is a double-edged sword. So for example, we have therapeutic treatments that may reduce the toxic effects and symptoms such as pain of the inflammatory response, but they may compromise the effective host's protection from the infection. Models that provide predictive power in cases like the complex regulatory framework involved in the inflammatory response can allow us insight into how drug therapies and dosage impact the cell mechanisms and consequently the system-wide response level. So with that, I want to specifically thank our advisors, Dr. Ramkrishna at Purdue and Dr. Subramaniam at UCSD, as well as Dr. Shakti Gupta and Dr. Mano Moria, shown here in this image, for their insight and contributions to the project. And I want to especially take a moment to acknowledge generous funding and support that we received through the Center for Science of Information. Here's a link to the paper that we published on the preliminary model that I presented. And I want to thank everyone here for their time. And if you have any questions, feel free to drop them in the chat box, and I'll do my best to answer them.
Thanks very much, Lina and Rubesh. Any questions? You could also press down the space bar on your computer if you have a laptop or keyboard and speak your question as well. Well, Lina and Rubesh, I know, especially Lina, you've been involved in this collaboration between Dr. Ramkrishna's lab and Shankar's lab for much of your PhD work. And I was just curious if you could just speak a little bit to the experience as a graduate student, kind of bridging between disciplines, domain areas, and how that affected the way that you approached your research.
That's a really good question. Thanks, Brent. So it's definitely been a unique opportunity that I really enjoyed being able to participate in. And I'm really grateful for the flexibility that Professor Ramkrishna has allotted me in order to be able to come out here to UCSD and work with the collaborators in Professor Subramaniam's group. It's definitely provided me a lot of insight into some of the biology, because the expertise that the Subramaniam group brings to this modeling framework is really just more insight into like what's really happening on a biological level. And at Purdue in the Ramkrishna group, Professor Ramkrishna, he provides a lot of insight into the cybernetic framework and just ensuring that we're honoring those, the framework that was initially established around enzyme regulation and ensuring that we define the system appropriately so that we have an effective model to describe the system. And so there have just been a lot of people that have collaborated on the project, both at Purdue with Rubesh joining, and we have a new Ph.D. candidate that's joining on the project as well. And so it's created quite a interactive environment on the project, which is really nice.
Thanks, that's terrific to hear. I know it's certainly one of the hopes for the center is to be a catalyst for helping to you know, bring people together like that. So that's terrific to hear. And it's also great that you're soon to be finishing your doctorate in this into the summer. So thanks again so much for presenting today. Are there any other questions at this time?
Brent, this is Shankar. If I may make a comment, I would, with your permission. The first thing I want to say is that, as you well know, I mean, I've been involved in CSOI from day minus 1 year. I think Wojtek, who is also on the call, myself and Anant, schemed and planned on this whole concept of science of information. But the main thing I wanted to say here is how, in the biological systems, living systems, how, science of information is crucial, and what this specific project, how does it relate to the larger framework of information? The first thing I want to mention is that when we look at complex mechanisms of physiology at the level of cells and tissues, for example, Lina pointed out to Rubesh and Lina pointed out to infection and to how macrophage cells respond to infection with the different agents. The concept here is that we can measure, with increasing technologies, we can measure a large number of things. However, we cannot measure everything because there are millions and millions of events that happen in cells that give rise to a given phenotype. For example, in this case, give rise to inflammatory response. So one of the questions that Professor Ramkrishna and I discussed years ago is the notion that if we can measure a part of the information or part of the data that pertains to this phenotypic response, can we infer the other response or the regulatory mechanisms that lead to this response through models such as cybernetic modeling approaches, which will help us in terms of if we know an objective function, which will help us figure out what is the right regulatory mechanism that is engaged, and then probably provide some insights, biological insights into this regulatory mechanism. That is the scope of how we started this project, years ago, just to give a perspective. Having said that, I want to say that the way we are thinking about the future, if you want to use methods like this, is clearly we have to have a well-defined notion of what is an objective function in these biological systems. In the case of project that Lina and Rubesh are highlighting, the objective function we are thinking about, an objective phenotype we're thinking about is inflammation and immune response. So therefore, any objective function that characterizes immune response inflammation would be a good objective function for us to use. In fact, Lina is using both cytokines and chemokines as objective functions, in this case, which we can measure. What about more complex situations where you have, for example, response of a cell, cancer cell to a drug treatment, or response of a lung epithelial cell to infection by COVID-19? So what would be the objective function? So this is a big question in biology. Can we decide what are objective functions? And if you know what the objective functions are, can we model the regulatory framework or regulatory network using what we can measure and inferring the other mechanisms which are operative in terms of giving the response. And so this is the global, I just want to place the context of the global framework of what we're thinking about. And there are cases where there may not be one objective function, there may be multiple objective functions which may concurrently work towards yielding a given response. And so then we have to look at the multiple objective functions. Can we define these? So one of the key goals of methods like this, modeling methods like this, is to test objective functions. We may not know the exact objective function, but to test and explore if one or more combination of objective functions would be adequate for us to infer the regulatory mechanisms associated with physiological function. And so this is where we are going in terms of the science of information to infer what is missing in this process. And I'm sure Shannon would be very appreciative, because this is really, the goal of what Shannon's hypothesis was how much can we really attribute veracity to information that is passed from an input to an output channel, and so how faithfully can we reproduce it? So this is the kind of question that we're asking through the objective functions. So I wanted really to have everyone have this perspective that Lina and Rubesh are working on. Happy to have more discussions on this.
Excellent, thank you very much. Any other feedback or comments or questions for Lina and Rubesh? Well, thanks again, both of you, for co-presenting today, and for everyone attending. We are recording basically the screen and the presentation that we'll have up hopefully next couple of days. So if you wanted to share it to other folks. And again, everybody stay safe and healthy, and we'll see you next time.
Thank you.
Thank you, Brent.
Thank you.
Yeah, thank you.